food and designing diets for these conditions. But I personally am a microbiologist, most interested in infection over the years, in the microbiome and in pharmabiotics. And so I just want to echo what Teresa just said about the importance of the microbiome. And I just want to give you three very short little 1 or 2 sentence stories about the microbiome to try and persuade you that this really is an important area for us. And the first thing that we should always remember is that we sometimes think we are the most evolved creatures on the planet. We have reached this high level of evolution, that we have technology and we can do all these things. But in fact, the most evolved organisms on the planet are, of course, the microbes. They've been here for 3.5 billion years. We've only been here for about 1 million years. And when we're long gone, they will still be here. So they're the most successful species on this planet, and they occupy every niche, including all the niches within us. So we only exist because microbes allow us to exist. So we're not the important ones, the microbes are the important ones. We just carry them around. If you want to understand the evolutionary importance of microbes in the gut, then all you have to consider is that in human milk, the mother is producing lots and lots of sugars that the baby simply cannot digest. And it might make you wonder, why is the mother in her human milk making so many things that her baby cannot take advantage of? And of course, what it is that the mother is already trying to create the right environment for a microbiome that will benefit her child. And so most of these sugars that the mother produces, these human milk oligosaccharides, can only be digested by the microbes in the gut. And so from birth, we have evolved to try and create a good environment for our bacteria in the gut. And lastly, we sometimes think, isn't it very odd, isn't it strange that bacteria produce serotonin and GABA and acetylcholine and transmitters and chemicals that we use in our brains? And we say, why do bacteria use these neurotransmitters? But of course, it's completely the other way around. As we evolved, we chose bacterial chemicals to use to make signals in our brain. But our bacteria still produce these molecules. So we share the same chemical language with our microbes. And when you share a language, you can communicate. I can't share your language of Spanish, but we can still communicate even with an interpreter. But bacteria and humans speak the same chemical language. And so it's not surprising then that there's a huge conversation going on between the microbes and the human. And if we believe this, if we believe that our health is actually a combination of our human aspect, our human genome, our human cells, our human organs, and these bacterial genes and these microbial genes and this microbial organ, which we essentially have within us, then health is an outcome of those two things working together. And of course, disease is when those two things break down. But if you think about it, we have very limited tools to impact on the human side of this human superorganism. We can use diet, we can use lifestyle changes, and we can use pharmaceuticals. But on the microbial side of our humanity, we can use far more things to influence the microbial side. We can use diet as well, of course, but we can use phage, antibiotics, prebiotics, probiotics that we heard about, symbiotics, pharmabiotics, fecal microbiota transplants. We have lots of options. And so as clinicians, you should certainly be thinking, sometimes maybe I don't need to treat the human side of my patient, I need to treat their microbial side in order to restore their health. And the therapeutic targets, now again, Teresa has largely introduced this, so I'll go through it very quickly, but you might think that the guts or the microbial members of our gut microbiome could maybe play a role in gut health, but maybe that's as far as it would go. But in fact, of course, what we have learned is the microbiome plays a role in skin health, in brain health, in lung health, urogenital health, even in cancer. They're particularly in cancer treatment, but also even in cancer the initiation of cancers, the microbiome can play a major role, and of course in inflammatory diseases. So the microbiome is in our gut, but it impacts our entire system. And what we believe in the APC Microbiome Ireland is that if the microbiome plays a very significant role in our health, then we should be able to mine things from the microbiome. We should be able to extract things from the microbiome and give them back to individuals in order to improve their overall health. So mining the microbiome for solutions. And as I said, normally I will be talking about the things above the line, the living things, the probiotics, microbial consortia, fecal microbiota transplants, and all of these have good evidence to support them that they have a beneficial role. But today I'm going to really talk about the pharmabiotics, the bit below the line, the inert, the inactive, although inactive is a bad word, of course, but they're non-living things that you can mine from the microbiome, like bacteriocins, bacteriophage, biomass, bioactives. Luckily for me, they all start with a B, so there's a nice synergy to that. But I'm talking about these things that you can mine from the gut. And the point I want to make, excuse me, this is causing some problems. Hang on. Okay. What I want us to appreciate is that life and health and disease and everything else is about molecules. A molecule doesn't know whether it was produced by a bacteria or a human, whether the thing is alive or not. A molecule is just a molecule, just like a pharmaceutical molecule is a molecule. And every biological phenomenon, me standing here and speaking, you listening, everything is mediated by molecules. And so what we need to understand is which of these molecules produced in the microbiome interact with which molecules in the human to cause health and disease effects. Microbes can only affect a host if they produce a molecule which can interact with a human receptor for that molecule and therefore cause an effect. And they could be produced prior to administration. It may be that some of these molecules are already present in the probiotic when you ingest it, or it could be that some of them are produced after you ingest the probiotic or the fecal microbiota transplant. And so when we think about it, and we've written a paper on this relatively recently, we have microbes, but then we have the microbial molecules, the effector molecules, the things which actually do something. And we can break those microbial molecules down, these pharmabiotics, into things like bacterial metabolites, maybe short-chain fatty acids, bacterial structures, the cell walls, peptidoglycan, lipopolysaccharide. It always surprises me, but maybe it shouldn't, that lots of the immune cells in the human gut, lots of the receptors on the surface of the immune cells are there to detect microbial molecules. They want to detect peptidoglycan, they want to detect lipopolysaccharide. So we're constantly communicating with our gut through our immune system. Maybe just bacterial biomass, just bacteria, whether they're alive or dead, can have an impact on our immune systems, on inflammation, on health benefits. And then there are things like enzymes and bacteriocins and bacteriophages. And I'm going to start at the bottom, these bacteriophages. Teresa introduced, or I should just say first that complexity is inevitable. This is not simple. This is not easy science. You might think I'm just using that as an excuse, but it's not easy. We're not looking at what you would look at in medicine normally, where you introduce one bioactive molecule, a pharmaceutical drug, to a system, and you just have to judge the impact of that one molecule. Here, if you introduce, let's say, bacteria, whether they're alive or dead, you're introducing millions of different molecules, and they're interacting with thousands of different receptors, and they could have multiple effects. So it's very complicated, and we need to understand that. Solving these mechanisms is difficult, and we're only at the beginning of solving these mechanisms. And usually, when we're trying to solve mechanisms, we don't do that in humans, because humans are not test subjects for testing mechanisms. We use animals normally. And In a perfect world, we would be able to show this probiotic produces this molecule, and this molecule is responsible for the complete health effect, and now we can just make that molecule and make a drug. That would be a perfect world, maybe. I just want to point out, Patrice already introduced this, that the microbiome is much more than just simply the bacteriome. Most people, when they talk about the microbiome, they're actually only talking about the bacteria. But we also have the archaea, these ancient bacteria which live in our gut, completely different kingdom from the normal bacteria. We have fungi, yeasts and molds. We have protista. We have phage, these bacteriophages that I'm going to speak about. And we have viruses as well as the bacteria. So it's very, very complex. On the right-hand side of the screen there, you can see the amount of bacteriophages, bacterial viruses in feces. So this is not concentrated. This is just the amount of bacteriophage you can see in a fecal sample. Now, bacteriophage are very interesting biological entities. They're not alive, but they do cause infection. They infect bacteria. And they do it in the three-step process that you see there. They bind, they inject their nucleic acid, usually DNA, and then that DNA hijacks the cell. It takes over the microbial cell and starts to just make more and more copies of the original bacteriophage, and then it programs the cell to burst using an enzyme called a lysin, and the cell bursts and releases lots of new phage, maybe 200 to 300 new phage for every cycle. And if you look on the right there, you can see a bacteria